Good evening, dear colleagues. I would like to present you Edgar, a deep learning-based program for prediction of folding energy of nucleic acids. RNA and single-stranded DNA molecules play a significant role in a number of biological processes. RNA secondary structure is especially important for understanding RNA functioning. Data on secondary structures is obtained using physical methods and biochemical methods. Both groups have their limitations. Physical methods are accurate, but quite costly and time-consuming, while biochemical methods can provide complete information about the structure at once and require application of prediction methods. Speaking of the prediction methods, there are three main groups: homology-based and knowledge-based methods, as well as methods based on predicted free energy minimization. The current methods also have their limitations, such as limited accuracy and performance. More so, most of them can't predict pseudonodes, which are essential in some structures. For that reason, most of the data obtained by biochemical methods contains errors. This is why a method of determining a structure optimality using folding energy assessment is necessary. That being said, we have developed a universal method for prediction of single-stranded nucleic acids folding energy. We implemented it in the form of Edgar program, a graph-based neural network. We began from gathering the RNA and DNA secondary structures data. The whole set was split into families. Then, these nucleic acids tertiary structures were obtained using the Rosetta software and molecular dynamics. We split the structures into separate fragments using a method, which is shown on the slide. To calculate free energy changes, we used the XTB program and GFN force field. Now, let's have a look at Edgar architecture. A nucleic acid structure is represented as a reduced graph, where the nucleotides are vertices connected by two types of edges, representing covalent bonds and complementary bonds. This information is passed to the neural network as embeddings layer. Then, Fourier positional encoding is used. It is later combined with the embeddings by summing the vectors. This makes the network know exactly which nucleotides form complementary bonds. To obtain global information about the whole structure of a nucleic acid, the resulting embeddings are processed by the transformer layer, which uses an attention mechanism. Besides predicting the target value, Edgar is also able to calculate its own uncertainty, which is proportional to prediction dispersion. Thus, the neural network predicts the folding energy and its potential dispersion based on the information about the primary and secondary structure. The model quality was assessed on a test dataset. The data obtained from molecular mechanics calculations was compared to the predicted values. Determination coefficient and RMSD were used as metrics. With a full dataset, the model R-square was 0.96 and its RMSD was 36.5 kilocalories by mole. One of the distinct advantages of the model is fast inference time compared to complete tertiary structure modeling methods. Edgar calculates the energy within 50 milliseconds for a 512 nucleotide structure and within 10 milliseconds for a 100 nucleotide sequence. The secondary structures data obtained by
You have 1 minute.
Good. The secondary structure data obtained by biophysical and biochemical methods was evaluated using Edgar. To do so, for each structure from the datasets, prediction was conducted using RNA fold method. Then, energy values obtained using Edgar were compared between the reference structures and the corresponding RNA fold predictions. The number of more energetically favorable structure within the reference data is shown on the slide. For the biochemical methods, the number of favorable structures did not exceed 12%, and for biophysical methods, it is more than 65%. This additionally confirms the statement about the high number of errors in biochemical structure data. So, in conclusion, the Edgar program is a fast and accurate method of determining the folding energy of secondary structures. This is a powerful tool for researching single-stranded RNA and DNA structures, which can increase the quality of software which processes structural sequences and data. Well, so that's about all, and thank you for your attention.
Thank you for your presentation. Now there is a time for questions. Professor Paroykov asks you how your study will help in computer-aided drug discovery. What do you think?
One of the most important things in drug discovery is to find out if the structure we receive is optimal. This is where Edgar comes in handy. Also, it will help people to study RNA structures, especially in case with aptamers discovery, which can be used both as drugs and delivery systems. Well, as you probably know, Celix efficiency is about 30%, and aptamers can substantially increase its efficiency. This is where Edgar will come in handy to evaluate the received structures.
Dr. Shulka asks you, how transferable is the model to the motifs not represented in the training set?
Well, most of the structures were received from PDB, but this information is still not enough. That's why we have decided to model it. And also, generally speaking, Edgar can also work with non-canonical pairs. Well, if that matters, just in case.
Okay. Dr. Yaroslav Paletrov asks, say you, Ivan, good prepared report. How much time needs Edgar to predict, for example, 100 mRNA of the about 1,000 nucleotide structures?
Well, we haven't tested every last one of the possible sequences, as you know. But anyway, we're talking milliseconds or seconds here. As you see, 512 nucleotides are calculated within 50 milliseconds. So, well, the inference time is kind of neglectable. It's going to be seconds anyway.
Okay. Dr. Yefremov asks, if I understood correctly, in the training set, 3D structures were predicted, not experimentally obtained. Why you did not use the later?
Well, we actually did use the experimentally predicted structures. They were used as a reference structures. So we compared, we used Edgar to evaluate the experimentally received structures and then compared with them with our predictions. So we actually did use them.
Another question is from the same Dr. Yefremov, comparison with known methods of SS prediction would be useful.
Yes, that's true. We did some comparison with the MXFold2 MFE methods, which is minimization of free energy. And the data, there is correlation between our data and what we received from MXFold, but any other MFE methods accumulate the error whenever the sequence length is increased. It's not the case for Edgar.
Okay, Vladimir, Professor Sulimov asked, what model of water is used in Edgar?
It is TIP3P.
Thank you. Is there any questions more? I think there is no question, so thank you very much once again for your very nice, interesting presentation.